Hey, welcome back. Okay, so this is just picking up from the last video. We're going to be finishing up the nervous system stuff we're going to start with synaptic transfer and synapses, move into neurotransmitters, greater potentials, and then go through some of the extra content. Okay, so the way that nerves actually transmit the electrical signals and transfer them to another nerve or another part of the body is through synapses. Okay, so if you don't have, so this, first off, this is actually really important to understand. Electrical signals are not able to be conducted if you don't have a membrane. Okay, so if you've got a bit of tissue that doesn't have the semi-permeable membrane, this phospholipid bilayer, it's the cell membrane, it doesn't work. Okay, so synapses are the physical gaps that separate neurons from other cells. So this is kind of what it looks like. Okay, so don't get freaked out by this yet. But this is what, so here's what we're saying. This is the end of an axon. Not necessarily, but we'll say in this example, this is the end of an axon. There's a little bit of space, and then there's a receptor, maybe a dendrite. It might be actually on the soma, like I said. This one might be a different, like a body tissue cell. This could be like a muscle cell or something. Okay, but so the way that synapses work, because they go through this really quick, okay, is very similar to the way the action potential worked in a axon or any normal thing. Once we get to a synapse, okay, the action potential is moving down the membrane like we saw in the last video. And what starts to happen is that the voltage change activates a different kind of ion channel. So we've seen it activate sodium channels, we've seen it activate potassium channels. Okay, once it gets to the synapse, it opens calcium channels, okay, Ca2 plus. Calcium comes in from the outside, it floods in, and what it does is that it signals to these vesicles here that we gotta go. Like, it's time, get the hell out, okay? The vesicles then move down to the actual synapse right here, right, the membrane right in front of the synaptic cleft. And these vesicles are full of neurotransmitters, which are tiny proteins or lipids, little things that will go and send a signal on the other side. Okay, so the vesicles bind to the membrane, the neurotransmitters flood out into the synaptic cleft, and then they will be received by these little receptor proteins, and those proteins will then send a new signal down through the next neuron or the next cell or whatever it is. Okay, so neurotransmitters is how we actually like feel things, and different neurotransmitters send different signals in different places. So, as an example, dopamine is a neurotransmitter that is generally associated with feeling happy or feeling good or feeling okay. Um, and one of the examples that we like to use, or that I like to use sometimes, is that like people with ADHD often have trouble getting enough dopamine in their system. So that's why very often people with ADHD are often looking for ways so that they can feel better or feel good and get that kind of dopamine rush. So that might be eating, that might be sports, or whatever it might be. These are your neurotransmitters. Okay. Sorry, actually skipped ahead of myself. There we go. So, neurotransmitters. So, like I just said, these are the chemical messengers that are released from neurons. They function to transmit signals across the synaptic cleft. Okay, we talked about that just a second ago. So, neurotransmitters can trigger different responses in different cells. Okay, so if the target cell is a neuron, so if at the end of the axon, this is a neuron, okay, we can see stimulation or inhibition. So that's a really important thing. Neurotransmitters can stimulate a signal to keep going, or they can actually be inhibitory. So they can stop a signal. They can tell a signal, don't move any further, we're done. Okay, so, but if the target cell is a neuron, it can stimulate or inhibit a new electrical signal, okay, to keep the signal going. If it's a glandular cell, meaning like your pancreas or your adrenal glands or something like that, stimulates or inhibits secretion. Okay, so one of the things that makes you like the one of the primary things that makes you like get that fight or flight response, that like pump of adrenaline whenever somebody spooks you, is because these neurotransmitters are coming in and they are sending signals to stimulate secretion of adrenaline, of epinephrine, so that you are ready to go. Okay. Or if the target cell is a muscle fiber, it can stimulate or inhibit muscular contraction or relaxation or extension. Okay. So an example. Okay, secretion, reabsorption of acetylcholine. So one of the great examples of neurotransmitters is acetylcholine. It's commonly released at muscular junctions, and it triggers muscle contraction. Okay, so acetylcholine is going to help you pump that iron. Okay, it's also commonly released within the autonomic nervous system to promote parasympathetic responses. So these are going to be the things that you don't actively do: breathing, digesting, moving things through your body, all of that. The acetylcholine is going to be doing a lot of that. So I don't think you need to know all that much about the receptor or the neurotransmitter itself. Like, you don't need to know all that much about acetylcholine, um, but you do need to know this. So acetylcholine activates the postsynaptic cell, whatever it might be. Remember that it's not just a nerve; it could also be a gland, it could be a muscle, or lots of different things. By binding to one of two classes of receptor: nicotinic or muscarinic. Okay, has to be continually removed from the synapse. Overstimulation can lead to fatal convulsions and paralysis. So if you get too much acetylcholine in your muscles, seizures. Okay. Um, yeah, so acetylcholine is broken down in the synaptic cleft into its two component parts by this enzyme, acetylcholinesterase. Okay, break that down. It's not that hard. Acetylcholine right here, really easy. That's its target. Esterase, the ester bond in the acetylcholine is going to be broken by this right here. The ACE means it's an enzyme. So acetylcholine ester ACE, it's going to be breaking that ester bond. So it's either released into the synapse from the presynaptic neuron, or it's embedded on the membrane of the postsynaptic cell. So it's either like as the acetylcholine is coming through, this is coming right after it, or with the receptor cell, it's going to be like sitting here ready to break it down if it goes. Okay. And so it will break it down into choline and acetate. So you can see that right here. Okay. So acetylcholine gets sent down, locks onto the receptor. It then goes through acetylcholinesterase, gets broken down by that into acetate into choline. The choline gets taken back up. An enzyme makes acetyl-CoA from the mitochondrion, by the way, and that puts it back together into acetylcholine, and on and on and on and on and on. Okay. Now, we did talk in ecology, my SL class. We talked in ecology about pesticides, and how, there are certain, when we talked about BT corn, if you remember, if you go way back, we talked about BT corn whenever we were talking about GMOs. Pesticides, a lot of them, the way that they work, they are neonicotinoid pesticides. And what those do is that they bind to acetylcholine receptors, and that's how they kill the insects. So let's talk about that. So neonicotinoid pesticides, what they're able to do is they bind to those nicotinic acetylcholine receptors, and they don't let go, ever. They never let go. So those pesticides can't be broken down by acetylcholinesterase, which means that they're constantly overstimulating the target cells. Okay, so insects have a different composition, and these acetylcholine receptors bind to neonicotinoids, which is the pesticides, much more strongly. They're more toxic. It can cause, so when I say it causes convulsions, it can cause like seizures. It can also cause like muscles to just completely lock up, and make it so that the insects just straight up don't eat or can't move, or just aren't able to do what they need to do in order to survive. So neonicotinoid use has been linked to a reduction in honeybee populations, which is not good. Okay, it's also been linked to a reduction in bird populations since the insects are dying off. So certain countries have restricted the use to be only so much. Okay, and that's what I've talked about before. And you can ask me, you can DM me or whatever. When I was working with honeybees, um, in college, this is the kind of thing we would do: is we would test these different neonicotinoid pesticides to make sure that they weren't having too much of an effect on the bees, or we would be testing different alternatives, which is actually really cool. And here's a picture of the kind of process. Okay, so in normal neurotransmission, the acetylcholine goes in, it binds, acetylcholinesterase breaks it down, and it gets reuptake right here. With toxicity, okay, neonicotinoids, they stop acetylcholine reception, and instead, these neonicotinoids, they're the ones that bind, and they irreversibly bind, so that that neuron is constantly firing a certain way and leads to paralysis, which kills the bug. Okay? All right. And last of the core content here, graded potentials. So nerve impulse, like I said, only happens if the threshold potential is reached. That was that first little bump before it shot up. Okay? So this comes in because of these neurotransmitters hitting these neuroreceptors. They will open ligand-gated ion channels, and it will cause these little changes that are called the graded potentials. So the whole way that a signal starts is neurotransmitter gets released, okay? That causes a slight change in voltage. That opens up a channel, which starts the action potential, and there it goes, okay? But this is what I was talking about. So these can either excite or they can inhibit. So excitatory neurotransmitters, so noradrenaline, they cause depolarization. Inhibitory neurotransmitters, GABA, they cause hyperpolarization, meaning that they actually make it so that the resting potential is even more imbalanced to prevent depolarization from happening. So combining the action of all the different neurotransmitters, lots of things different happen. If overall there's more depolarization, then the neuron will fire. If overall there's more hyperpolarization, the neuron will not. So here's your example. So if we have these different neurons, one excitatory, one inhibitory, okay, and we are able to, we wouldn't be able to do this, but if we were able to plug in and see what our different potentials or different voltages look like, well, so we can see that here, this one is giving some depolarization, okay? Here, this one is showing hyperpolarization. Overall, it just needs to get over the threshold. If that happens, it'll send an action potential, okay? And that is pretty much it. So I'm going to leave this one here. I'm going to let you guys look at extra content if you want to. And like I said in the last video, I will be uploading these with some more supplementary materials, including the mouse party simulation, which I suggest everybody do. It's a really important, really cool way of figuring out neurotransmitters. So thanks a bunch for watching, guys. Until next time, Gucci.